What if you could make a genetic copy of a living being? Not a look-alike, not a twin, but a true biological duplicate. Sounds like science fiction, but cloning is very real. So, what in genes is cloning really doing? Welcome back to my channel where we unravel life's biggest mysteries written in your DNA. Today we're diving into cloning. What is cloning? Cloning is the process of creating genetically identical copies of DNA, cells, or even entire organisms. The key word here is genetically. Same DNA sequence, same genetic blueprint. But cloning isn't just one thing. It comes in very different forms, with very different goals. Let's talk about organismal cloning. This is the kind most people think about. The most famous example? Dolly the sheep. She was created using a technique called somatic cell nuclear transfer. Here's how it works. Scientists remove the nucleus from an egg cell. They insert the nucleus from a normal body cell. The egg is stimulated to start dividing. The embryo is implanted into a surrogate. If it works, the result is a new organism with the same nuclear DNA as the donor. Sheep, cows, goats, pigs, cats, dogs, even monkeys have been cloned this way. But here's the catch. Cloning is extremely inefficient. Most embryos fail to develop, often due to epigenetic reprogramming errors, not DNA mistakes. Next one is embryo splitting. This method is much simpler and happens naturally. Early embryos can be split into separate cells, each capable of becoming a full organism. That's exactly how identical twins are formed. Scientists can do this in the lab too, mainly in animals and livestock breeding. Same genome, different individuals. Therapeutic cloning is where cloning turns medical. Therapeutic cloning also uses SCNT, but the goal is not to create a living organism. Instead, scientists stop the process early to harvest embryonic stem cells. Why? Because these cells can model diseases, test drugs, and potentially regenerate damaged tissues. Since the cells match the patient's DNA, the risk of immune rejection is much lower. Still, ethical debates remain, especially because embryos are destroyed in the process. In molecular biology, cloning doesn't mean animals at all. It means copying DNA fragments. A gene is inserted into a plasmid, placed into bacteria, and the bacteria replicate it millions of times. This is how we study genes, produce insulin, make vaccines, and engineer proteins. It's cloning at the molecular level, and it's everywhere in modern biology. Are clones perfect copies? Genetically, yes. Biologically, not always. Clones often suffer from developmental abnormalities, organ defects, early aging, and high mortality rates. Why? Because cloning struggles to reset epigenetic marks, the chemical switches that control gene expression. The DNA is the same, but the instructions aren't always read correctly. Cloning isn't just about science, it's about survival. Endangered species like black-footed ferrets, Przewalski's horses, and mouflons have been cloned to restore lost genetic diversity. Using frozen cells and surrogate mothers, even from related species, scientists can bring back genetic lines that would otherwise disappear forever. Still, cloning is not a replacement for habitat protection. It's a last resort tool, not a magic fix. What about human cloning? Human reproductive cloning is not practiced. Not because it's impossible, but because it raises massive ethical, medical, and societal concerns: identity, consent, health risks, psychological impact. Therapeutic cloning is debated. Reproductive cloning is broadly rejected. Cloning shows us something profound. Genes can be copied, life can be repeated. But development is not just DNA. Epigenetics, environment, and chance still shape who or what we become. So cloning isn't copy-paste life. It's more like copying the code and hoping the system runs. Next time you hear clone, remember, same genes does not mean same destiny. Until next time, see you soon.